devastating disease where your own body's immunity attacks your own body, your own body's immunity attacks your own joints. To be able to come up with a better treatment than what currently exists, we have to understand the underlying mechanism and the cause of the disease. And it turns out the subset of cells of your immune system are these T lymphocytes. And T lymphocytes have already been shown and validated to play a major role in this process in rheumatoid arthritis and a lot of other autoimmune diseases. What pharmaceuticals do usually, they take cells like T lymphocytes and they throw about a couple million compounds at them, and then they look at the effects that they want to see. The problem is that cells as a whole are complex systems. So you're going to see the effects that you want to see from the molecules that you throw at them. But then those molecules are doing a bunch of other things that you don't really see. What we wanted to do was to go a couple of steps further and identify the molecular mechanism of the process and really figure out what the root of the problem is, what are the root entities of the problem is, and then target those guys. So what I'm going to talk about is a couple of proteins. It's going to be STIM and ORAI. There are two major proteins that play a major role in this process of T lymphocyte activation and also rheumatoid arthritis. I'm going to focus on ORAI, which is an ion channel. It's a calcium ion channel. And it needs to be activated by binding directly to the STIM, which is the blue protein. So STIM has to come and bind directly to ORAI as a channel to activate it. And you get a calcium influx, and that calcium influx is vital for T lymphocytes to be activated. We wanted to take this even one more step further and not just target the protein. We wanted to target pieces of the protein that are in charge of this process. So that way we thought we could actually derive more specificity, less toxicity, and less side effects. So instead of going after the whole cell, we want to go after the piece of the proteins that are in charge of this process. So we think we have three major advantages in this case. One is basically the emerging target. ORAI by itself, the channel has been discovered in the past few years. It's been validated and shown to be a major player of this process. And it's also a nice target because there are patients who have natural mutations in ORAI, and those are the bubble boys of 80s. They seem to have no side effects. They do fine in daily activities. The only problem that they have is their body's immunity is compromised. So that's why ORAI makes a nice target. The second advantage that we think we have over the market is our unique strategy. Instead of after going a complex cell, we're going after pieces of proteins, not even a whole protein, pieces of proteins that are in charge of this process. So we think we can get a specificity and much less toxicity and side effects. And the third advantage that we have is basically the library of compounds that we used here to identify our drug targets or drug candidates. Generally, pharmaceuticals buy their compounds from the same sources who buy their compounds from the same sources, and for some unknown reason, the original source always is the previous Soviet Union. And in this case, we collaborated with MIT and Harvard, and we used a library that was homegrown at the university scale, and also there were compounds that were natural products that were part of this library. So we think we have a unique library of compounds that's kind of orthogonal to what pharmaceuticals use. So how did we actually identify the small molecule drugs, the drug candidates that we have? So I told you we have identified specific domains, specific pieces of the protein that are in charge of this whole process of rheumatoid arthritis. What we did, we just did a simple binding assay. We took 12,000 compounds, and we took these pieces of these proteins, and we just did a binding assay. And whichever molecule bound tightly to these pieces of proteins, we took as a hit. And the hypothesis was, if we have the ORAI, and we have a small molecule, we have a compound that binds to the important pieces of ORAI, it's going to somehow perturb the function of ORAI, and it's also going to prevent it from binding to the other protein STIM and kill the activity. And that was the hypothesis. So we have a target, we have molecules that bind to the target. Now, do they actually work biologically? The first thing we wanted to look at was calcium influx. ORAI is a channel, it's a calcium channel. We want to look at the calcium influx through the channels. And that's what we did with calcium imaging. There's a few compounds here that we have tested. What I want you to focus on is the black trace and the red trace. The black trace is just the control. It's non-compound treated. And you can see the difference with the red arrow. And the red trace is compound treated. The calcium influx between the control and the compound treated has significantly been reduced. And that calcium influx is vital for T lymphocytes to be activated. Without that calcium influx, T lymphocytes would not be activated. The second experiment that we did was a cell-based assay. We looked at 83 genes that would be upregulated, overactivated when T lymphocytes are activated. Each bar here represents one gene that's activated when T lymphocytes are activated. So if you take a T lymphocyte and activate it, all of those bars should be above the plane. After treatment of the compound, as you can see, most of the 83 genes, they have been downregulated, some of which have been downregulated 150 times. So this shows that the hits that came out of the library not only bind to the target specifically, they also inhibit T lymphocytes from getting activated. The next thing we wanted to do, what is the effective concentration of the compound, the hit that came out of the library? And it seems to be about a little bit less than 1 micromolar, which is pretty good for original hit. We like it to be about lower nanomolars, but as a starting point, it's not that. So just to summarize what I told you so far in these couple of slides, we have a target, we have a specific target instead of going after a complex cell. We have molecules that bind to the target, they perturb the function and activity, and they reduce, they downregulate the genes that are affiliated with the activation of T lymphocytes and with the disease. So the last piece of data I want to show you guys is our in vivo models. So we're working on a few in vivo models. The data I'm going to show you today is very preliminary. It's coming out of just the T lymphocyte model. And generally in the T lymphocyte activation models, what happens is you take the mice, these experiments are done in mice, and you expose it to a foreign body, like an antigen or anything that T lymphocytes would recognize as a foreign body. They get sensitized, and at a later time point, you come and challenge those, so you expose them again to the same type of foreign bodies, and then T lymphocytes get activated. Meanwhile, you treat the mice with the compounds of interest and see what kind of effects you get. So here's the data. We have, in this case, we have three groups of mice. The first one is the PBS, which is the positive control. And what we're looking at here is just IL-2, which is a gene. It's a hallmark of T lymphocyte activation. And if you see bright bands up there, that means T lymphocyte is super activated. You can see the quantification on the other side, which is about 100%. So in the PBS, where there's no compound treatment, you have full activation of T lymphocytes. The second group that we have here is cyclosporin A, which is a known commercially available immunosuppressant. And as expected, when you treat the mice with cyclosporin A, you get faint bands, so T lymphocytes are not as activated as in the control. And the third and the most important piece of data here is the mice that were treated with our compound that came out of the screen. And it seems to be comparable as far as T lymphocyte activation, seems to be comparable with the commercially available cyclosporin. And this is just the original hit that came out of the library. We haven't done any medchem or anything around this. Before I move on to KB to talk about the marketing, I just want to touch up on the IP real quick. The methodology and the compounds have all been developed